So now our second speaker of this session is James Reecy, who currently serves as Associate Vice President for Research, where he oversees the Office of Sponsored Programs Administration and internal funding programs, and fosters the development of interdisciplinary teams at Iowa State University. Dr. Reecy. So this is the...
Okay. I want to thank the organizers for inviting me to come here today and talk. And for full disclosure, in terms of trying to give you some background of where I come from, the first animal in which I owned that did go to slaughter was back in the 1970s. And my family is now in our at least fourth generation of livestock production in a tangential way, in that we really don't own the animals. We end up being part of the associated infrastructure for livestock production. And if you go back to the 1980s when I was going through college, my parents were like, do not go into agriculture. There is no money to be made there. And at that time, that was in the middle of the farm crisis. And I mean, a lot of people were going out of business. But being a young, stubborn individual, and my mother is probably as stubborn as they get, decided to stay within agriculture. But I went and did it more on the academic side, and I've been part of the university ever since. What I'd like to talk a little bit is really focusing on that in vitro cell culture or in vitro meat for the vast majority of it, and a lot of the science associated with it, because it's a lot of the science which I've done over my lifetime so far. And try and compare and contrast that with the meat industry itself. A bit of improvements of which agriculture has done over the last couple of decades, because we talked about that incremental innovation that has occurred. And this is an industry that is extremely innovative and has been continuously doing it. And then some of this will do a little bit similar to what Jan had done in terms of implications for climate, natural resource, some cultural considerations that we have to take into place for this, and then also nutritional concerns. In terms of that in vitro cell culture, we got to go back to what is the product in which it is that the consumer is taking. And it is muscle, very clearly. And it is that structure of that muscle that ultimately provides the taste in which it is that we are used to having. What is that mouthfeel? What is it that we expect in terms of doing? Within that muscle itself, in order to turn this into something that we could do, as Jan put it, in the bioreactor, we have to have a cell that is in place. And that cell, in this case, is this stem cell or satellite cell or myoblast. These are single nucleated cells that have the ability to replicate. So as you start in any young embryo, you got a single cell. What we are today is now multiple billions of cells, and you have to have that cellular replication. For muscle to be formed, you have to have these cells fuse to where we now have multiple nuclei in a single muscle fiber, and that's where the protein actually gets into. So this is the bioreactor individual cell that we have in a growing organism for doing stuff. But it's these cells that we would take out for the satellite cells that we would then use in a bioreactor external to the animal in order to produce meat. So anyone who says an animal does not get harmed in this scenario is outright lying to you. You still have to have a source of cells. Now, are there a whole lot less animals that would get harmed in terms of doing this? Absolutely, the case would be that. And I'll get into that a little bit later. But ultimately, you take these cells out, put them in culture, and then you have to ramp up that process in order to, in order to ultimately get enough in order to get to that $50 patty that we can do today or the $330,000 a few years ago. I truly believe that the system will work and we'll get it to a point where it is cost competitive with other protein sources that are out there. To me, that is a simple engineering problem, And I am being very glib about saying simple in that case because there are a lot of innovation that has to occur in order to allow that to happen. So in terms of these cells, and one of the reasons why I say that you will always have animals involved in this is today, any cell can only replicate so many times until you get to, in essence, senescence or a stationary phase. And that is because if we go into the cells that we have on the DNA, on the end, there's something called the telomere. And that is necessary in order to stabilize the DNA. And every time you replicate, it shrinks. Well, these satellite cells don't have telomerase, which would extend that telomere in order to allow you to grow indefinitely. So there's only so many times that they can replicate. Once you get to that senescence portion, you've got to start over with a new set of cells. Now, we could genetically modify these cells to where they would produce telomerase and then would be able to replicate for a greater extended period. But now you're dealing with something that is going to be a genetically modified organism. Now, if we genetically modify it after it's left the animal, does that have the same connotations as a genetically modified animal itself in terms of what people would accept? Because the animal may be perfectly normal in terms of what it is from a natural standpoint, but then we modify it after the fact. How would we handle that for our labeling and our other reporting in which it is that we have? So a little thought exercise in terms of doing this. And let's assume, and a lot of this is a lot of assumptions in everything in which I have here, is that we get about a little more than a half a kilogram of these satellite cells from an entire beef cow. So we take every bit of muscle that is there, we isolate these cells. It would take, excuse me, roughly about 9 doublings in order of that mass to get back to the same amount of meat that a single animal would produce today. But if we then got it to go out roughly 30 doublings, we could get almost 1.5 billion pounds of product from the equivalent of a single animal. Now, how far we can go, because Jan was right, how far can we go in terms of these doublings before those cells run out? Don't know, in terms of doing it. Anything in which we do in cell culture in my own lab, we only let the cells go to roughly probably, about 20 doublings or so. And it's out here at the end that you have that massive increase. So anything in which you change there is going to have dramatic impacts upon the amount of product that could be produced. If we look at this in terms, and I'm trying to put this into context for the beef cattle industry in the United States, there are roughly 39 million head a year produced. They all weigh, on average, somewhere close to 1,250 pounds, and you get about 525 pounds of meat out of that. You're now talking about 20.5 billion pounds a year. There's actually more in that occurs in the United States because we have imports into the United States as well. If you could get the number of 30 times for doubling with that 1.46, you're talking about 14 head per year to completely change the industry. If you could take 39 million head, move it down to 14, I mean, that's an absolute game changer. In order to do that, there is a phenomenal amount of innovation that would have to occur in order to make this a reality. But I'm taking this all the way to the extreme in terms of doing stuff, but to give you some idea. Now, what does the meat industry then look like? Essentially, it becomes one giant fermentation vat. And what is the innovation that you need in order to pull things in order to produce that product? Because when you look at this, there is talk that, okay, this could occur at a family unit in small bioreactors in your house. Food safety alone, I'm going to believe that that's probably a problem. But You could turn it into a large organization to where you now have a different form of a massive industrial complex in order to have the inputs and then the outputs and processing, in order to change that entire food system that you had from production through in terms of doing. The level of innovation that has to occur, and Jan alluded to this, how do you try and keep these cells, in essence, sterile so it stays just muscle? Bacteria love to grow, and they grow a lot faster than what any mammalian cell will grow in this exact same media. So keeping the bacteria out, but the muscle cells there, is much of that trick. And the innovation will have to come is how to do this without using massive amounts of antibiotics. We have a big interplay right now between what is human health and livestock production, but we both need antibiotics for that production to occur. This system, while the companies say, and we don't know how they would go about doing this for scaling it up, because as Jan told us, they've kept most of this information proprietary. They have to keep things sterile. And how they're going to do that, unknown yet at this point in time. So there are numerous startups, as Jan had alluded to, that are trying to innovate and work in this system. And it's really quite easy to understand why they would want to do so because of the size and scale of what it is that we have. Because if you look in the United States and worldwide, it is a massive place. So we'll switch a little bit now from what it is and how we produce or could produce cell culture-based meat, and compare and contrast that a little bit with more traditional livestock production. In both cases, we have our inputs and our outputs in terms of doing it. The livestock themselves, they are our bioreactor today. In the case of ruminants, taking grass, other roughages, as well as some concentrates, minerals, vitamins, and others, and then producing, and I only show the meat here, but there are numerous other products that come out of this process as well, and I'll talk about that somewhat later. One of those, which happens to be something that people tend not to like to talk about, of manure, but manure has phenomenal amounts of value because it is organic matter that can go back into the soil and recirculate portions. Similarly, you now have to, instead of having more roughages and concentrates, in this case in the form of whole plant cells, you now have to essentially have purified amino acids, glucose, that does have to come from some plant somewhere, still has to be produced, but it's maybe in a different form that then would go into the bioreactor to ultimately get here. This also has a lot of waste products from it, because all of the cell culture media that is put on this then also has to be processed out the backside, much like you think of it, of the waste that you have from any other processing plant. So in some respects, it's much like manure. The only difference is it's in a purely liquid form in terms of coming out of these plants. But you would have to deal with all of that as well. Swine production, poultry production, about the only difference in the whole system is you remove the roughages and you now just really have concentrates that are going into it, but slightly different products. In terms of protein production over time, to give you some context as to what has already and is occurring on a daily basis when it comes to livestock production. And as Jan had pointed out already, the exact same slide, trying to say, okay, what is the size? And it is this part right here where in the US alone, it's essentially $1 trillion of the impact. It's 5.6% of our GDP is meat in terms of what it means to the industry. It is huge. So it is very easy to see why any venture capitalist, any company is very interested in trying to change and really dramatically modify what is the system in which we have because of the money that is possible to be made. So in the dairy industry, and if we go back in this case, really to about the 1950s versus today, Milk production in the United States has continued to go up and is pretty much linear over the last couple of decades. So it's almost a doubling in terms of the amount of milk that is there. Versus the number of cows that we have has gone from roughly about 25 million down to not much more than 10 million. But that milk production per cow is just short of 4 times what it was. So today we have less than half the cows and we produce almost twice the amount of milk in which it is. So it has had a huge change on the environmental impact that livestock production has back in the 1950s versus where we are today. I could say the same thing as it pertains to beef or poultry. I mean, poultry, years ago, it essentially took 120 days to get a whole carcass in terms for what it would be for a bird that you would buy in the grocery store. Today, it takes 36 days. And that poultry animal is now more than twice the size of what it was back in the 1960s. So you want a massive increase in the efficiency of what has occurred is there. For CO2 emissions, while those emissions per animal have gone up, per unit, it's essentially almost about a third of what it was back in the 1960s because of that increased efficiency or unit that we have, or unit of milk per unit of animal. And the same thing would occur for poultry, beef, and pork as well. In terms of where we were and how this production is taking place, most of this increase is attributed to the genetics of the animals in which it is that we're working with. And if you go back here, and these are in different colors because of technological changes that have been implemented in those years. So prior to 2004, it was very much Mendelian genetics. We select the best animals that have been progeny tested to say what is the genetic potential of that animal. But it took years. Roughly took about 8 years to understand whether or not a bull was going to be a genetically superior bull or not. And at that rate, we were increasing the value of an animal, in this case a cow, is about $20 a year. So if a cow was born in 2017, it is $20 less than a cow that was born in 2018, and so on. So we were, in essence, increasing about $20 a year. Then in 2005, we started a process of genomic selection using the genotype of the animal to understand what was the genetic potential of the animal. And this was based upon the genotype of the sires. And in doing that, we essentially did a little bit more than a doubling of the rate at which that genetic improvement took place. And then in 2010, we essentially switched to using the cows themselves, because that's the vast majority of the animals we have, in order to put additional selection pressure on the cows. And in doing so, we essentially doubled the production efficiency or increase that we have as well that's taking place. And that continues on year after year. So in order to compete, the industry for in vitro is going to have to do the same thing. How are they going to sustainably keep increasing their efficiency in order to have the parity that is there? So in terms of climate and natural resource, I think I'm going to speed by most of this because Jan pretty much covered most of it. With the exception, I will put in this eutrophication potential. In this case, there's only been one life cycle analysis that's been done. And one of the unfortunate things that they did is they never put the spent media into what it is that they had. And even in doing that, okay, it's close to what poultry production is today. But by vast majority of all the amino acids, nitrogen is all coming out at this end in terms of doing stuff. So it's going to move that probably much closer to or maybe even exceed what it is that we have for potential production today. So from a climate standpoint, it could have a huge potential. We do have the concerns for the growth media. So we think about it for dairy cows today. People have problems of adding growth hormone to the cow in order to get more milk production. Well, in order to grow these cells, we're also adding hormones directly into this media. What will be the perception of the public for adding these hormones to these cells after they've left the animal in order to stimulate production as well? Cultural considerations. We have to also deal with ethnic diversities for cultural and halal. So how we actually would collect these cells at the day in which they come from the animal itself would have implications for how it is that you'd be able to market these products after the fact, because it is still derived directly from the animal itself. And then nutritional. There are a number of concerns in terms of doing this, because much of the nutritional value in which we have from traditional meat in terms of like heme iron and other things for the availability now have been removed because those cells are no longer present within the product in which you have. Now, there are opportunities because you could change the fat composition in cell culture to a much greater extent than what we'd be able to do in a live animal in terms of what we're producing. And then, as Jan had already talked about a lot before, is also the taste and feel and everything, the product in which you have, and I won't go over that. Now, the importance of the livestock industry as a whole, and this goes beyond really what we're talking about within the United States, and what is the impact really in the developing world. And I can go very squarely to the country of India, where they have, in essence, about, well, way more animals than what it is that we have. But it is that income, the nutrition, the rural vitality, Social Security, the leather, the wool, all of the other associated products that are coming out of here, that you're now changing where that portion of those products are going for other products, and what is the implications not just in the food chain, but in other commercial products simultaneously that would also have to undergo innovations if you remove the animal from the entire portion. Now, it can be done. I would believe that it could. But it does mean that you're impacting more than just one portion of society in terms of doing this. And for all of this, I have to acknowledge these four young ladies. They are undergraduates at Iowa State University. That this past summer, they spent about 2.5 months at the FAO in Rome, working very specifically on this project. And in reality, all of the citations and everything that I had throughout was thankfully that they helped to pull everything together. So they actually deserve all of the credit for everything in which I just relayed to you. And thank you very much.